This is another portion of a chapter that's going to be extremely long, and I'm just going to go ahead and warn you there's quite a few mini lectures that go with this as I try to break it up into, different distinct parts, to make it easier for you to follow along so you're not sitting there listening to me drone on for an hour. So let's talk disease. Transient microbes. You have your resident microbes. You're touching things. You're being exposed to things. You're being exposed to people. Right here now in the year 2020, COVID-19 is an issue. It's being spread. Some people are getting it. It's becoming part, it's a transient virus in their sinuses, but they're not getting sick. Other people, it's a transient sinus colonization that it's going to lead to a disease, lead to an infection, which is going to become a disease. That's the thing we're having an issue right now with COVID-19 is that the talking heads, the people on the news who have no background in science are trying to play scientist, and they're trying to get across certain things. Epidemiologists, people who don't have a background in microbiology or epidemiology are trying to figure out how to deal with the spread of COVID-19. What we see here is that they have mixed up what is a colonization, what is an infection, and what is an active disease. 98% of the people who test positive for COVID-19 don't get sick. They don't even know they have it until they went and got a swab. Ooh, I'm positive. Okay, that means that the virus is now colonized, part of their normal flora. It may be there for a while, a few days, a few weeks, a few months, maybe a couple of years, or it may be there permanently. It's now part of, it's going to be passed around, kind of like, influenza is nowadays, the common cold is nowadays, chicken pox, things like that, just going to be passed around. Small percentage of the people get an infection. Ooh, I got a slight fever. Oh, my nose is getting runny. I have a sinus infection. No big deal. Unfortunately, an even smaller percentage, 1 or 2% of the people who, test positive COVID-19 get a disease. Sinus infection becomes chest infection, lung infection, pneumonia. The pneumonia leads to pulmonary, cardiac. For some people, other underlying issues they had become more pronounced. So, in this portion of the chapter, we're going to look at, what it means to be colonized, what it means to have an infection, what it means to have a disease, what are pathogens, what are just part of your normal flora that are just there as your normal microbiome. Now, pathogenicity is a microbe. We like to think of bacteria mostly here when we think pathogens, is the potential that a microbe can cause harm. Some microbes, they're there, they may or may not cause an issue. One person has this microbe, it's there, it's part of the flora, it's a transient part of the flora, no problem. Another person, this transient microbe can become a pathogen, can cause an infection. So, there are true pathogens. If they're there, you're going to get sick, it's going to cause damage. Malaria, Plasmodium falciparum, Plasmodium malariae, and the other Plasmodium species. Once there, you've got malaria. There's no ways about it. Okay, just its normal life cycle in us is going to cause disease. Its mere presence. If our immune system can't wipe it out as soon as it's there, within a few days of it being there, before you even notice it, during the incubation period, well, you're gonna get malaria. Rabies. Oh, I love me some rabies. Rabies. If the rabies virus gets inside of you, you're gonna get rabies. There's no, am I gonna get sick? Am I gonna get disease? It gets in you, you have an infection, you have a disease, you will die. Done. At the other end of the spectrum, you have the opportunistic pathogens. They're there, for the most part, for most people, the majority of people out there in the world, it's actually even going to be part of their normal flora. Just under certain conditions, certain times, certain something, whatever. Phase of the moon, your diet, your stress levels, where you are in your monthly cycle, whatever. It's going to trigger these organisms to suddenly become pathogens. They're going to overgrow everything else, and they're going to cause an infection. Candida albicans, yeast infection. Guys, girls, both get these. The genital yeast infection is one that we think of most often. It's there right now. Whether you have a yeast infection or not, it's part of your normal flora. Something changes. Maybe it's your your personal hygiene for a few days is off because of whatever. For women who are more susceptible than men, for women it can be part of their cycle. For some women, just at certain points in their monthly cycle, they're going to get a yeast infection more often than not. Candida albicans suddenly just able to overgrow everything. Instead of being the one fan in, the one person in a, in a stadium full of other fans, it now can start to grow. It can take over. It can overgrow everything else. When it comes to pathogenicity is a yes, no. Can it cause damage? Yes, no. Virulence is to what degree? If it can be a pathogen, the virulence is a sliding scale. Low virulence means that this pathogen, minimal damage. Highly virulent means lots of damage, probably death. Rabies, true pathogen, because there's no in-betweens. If it's there, you're going to get sick. And it's highly virulent, meaning that you're going to die. Candida albicans, low pathogenicity, because potential is kind of there, not really. Virulence, very low. Because for most people, if you get a yeast infection, it's just irritating. The most damage you're going to get is the inflammatory response causing a rash. Now, as I mentioned before in a previous lecture, I believe in Chapter 10, Chapter 9, I also mentioned it. A lot of infections aren't going to be because just one microbe's there. Because think about it. You're covered head to toe in billions of bacteria. You're picking up thousands, if not millions, of transients every day. No place on your body is it just one type of microbe there and just one only. It's a mix of hundreds and different species. In your mouth, there's, who knows how many trillions of bacteria there. And there's, depending upon which paper you read, who you're talking about, the diet, the smoker, whatever, anywhere from 300 to 500 different species of bacteria. I ate a chip at lunch. It cut my gum. Microbes are getting in there. It's not just one microbe. It's, one microbial species. It's dozens, if not hundreds, of different species are getting in there. Not all of them are going to be able to establish. Not all of them are going to be able to cause infection. But we have to keep in mind, we get into this mindset, infection, Koch's postulates, one microbe, one infection. No. We're starting to see that infections, disease can be started off because of one microbe, but there may be a secondary, a third, or a fourth microbe that's also present at the site of infection that's also leading towards some damage. Now, when it comes to this colonization, to have the disease, you have to have an infection. To have an infection, you have to have colonization. Well, to have the colonization, the microbes have to get in you, get on you. So we have broken up disease and infection into steps, different steps that the microbes have to go through so that you have a disease. First step, step one, got to get in. You've got this dry outer layer of epidermis that you're constantly shedding. Heck, all the dust in your room at home, your dorm room. That dust, basically, it's the majority of it is your dead skin cells. You're constantly shedding. And as you're shedding those dead skin cells, you're shedding hundreds, if not thousands, of bacteria on them also. You have your mouth is constantly, you're salivating. It's a rinsing action. You're rinsing food. You're rinsing microbes down your mouth, out the back of your mouth, down your throat to your stomach. Constantly blinking, keeping your eyes moist, your corneas moist, but also you're wiping away, you're washing your corneas of any dust particles. But somehow things have to get on you, things have to get in you. Every microbe has a specific portal of entry. If this microbe, be it a bacteria, be it a protozoic parasite, if it's one that the disease is going to be associated with the large intestine, well, if I get it on my hand, it can't get from my hand to my stomach. To get to my gut, it's got to go through my mouth. Portal of entry. COVID, influenza, common cold, portal of entry. Your nose. The majority of STDs, the common portal of entry, urogenital tract. Certain skin infections. Well, based on what I just said, they got to be on your skin. Skin, mucous membranes lining your mouth, your eyes, your sinuses, your urogenital tract. Those are portal of entry of choice. So when you look at table 11.2 here above my head, portal of entries, skin, GI tract, respiratory tract, urogenital tract. Just a small listing of human associated microbes that will come in these different portals. How access is gained. Nick, a cut in the skin. There are certain bacterial microbes, certain bacteria that, are able to digest away the proteins that are holding the dead keratinized skin cells together. So they can make microscopic fissures that they can now penetrate down in through. Gastrointestinal tract, you're pretty much going to have to eat them, drink them. Because of the gastrointestinal tract, we came up with the term fecal oral. Fecal oral. You ingest some microbe, it gets into your stomach, small intestine, large intestine, colonizes, cause disease, and then you pass out some of those microbes, or if it's the parasitic worms, the eggs of the microbes out in your feces. That feces that's now contaminated with the bacteria, the parasite, or the parasitic eggs, then gets into the local water source, or is part of the fertilizer for foods, for food crops. Next person goes, drinks the water from the well that the sewage contaminated. Somebody goes and just picks some of the food, the fruit, or whatever off the ground in an area that they're using human waste or animal waste or water that has been contaminated with raw sewage, their fecal oral. Now, just because you've been exposed to a microbe, be it a bacteria, a virus, or whatever, just because you were exposed doesn't mean you're going to get an infection, doesn't mean you're going to get to the disease. How big was the exposure? There have been clinical studies where researchers want to know just how many does it take to cause an infection, just how many does it take to cause a disease. So they pay students. You'd be amazed what students, poor students, would do for $1. Now it's a few bucks, granted. They will go, and as you see below, this little partial list of some clinical studies they went through. E. coli 0157H7. This is the jack-in-the-box E. coli that caused people to get bloody diarrhea and die. They went and they paid students, paid anybody who wanted to come in and do it actually, but majority of the times it's students. Here is a vial of liquid. We have calculated and diluted out, so that we know roughly how many bacteria or how many CFUs, colony-forming units, are in each vial. Every person gets one vial. Chug it down. Keep track. Let us know if anything happens. So, using those types of studies, they found that 0157H7 takes less than 100 bacteria. 100 bacteria. That's next to nothing. Hepatitis A, they did the same thing. Hepatitis A, people get hepatitis, which is they can treat. They calculated it out, 10 to 100 viruses you have to ingest. Salmonella, the one that's going to really, Salmonella typhimurium is really going to cause you bloody diarrhea. Salmonella typhi is going to cause you copious amounts of diarrhea. Less than 10. The majority of E. coli strains that you're going to come in contact with, not the bloody diarrhea 0157H7, but the majority of the bacteria you're going to come in contact with contaminated meat, if the cow or the chicken or whatever was not butchered correctly, they punctured the large intestine and they didn't clean the carcass or whatever. 10 to the 6th, 10 to the 8th bacteria. That's 1 million to 100 million bacteria. Now, that's a large number, but, if you were to look at it as a pellet of bacteria, this would be a pellet of bacteria that's probably going to be in the size of a BB. If you were to grow a big thing of it, 100 million to 100 million, and to spin it down in a centrifuge, this would give you a pellet of bacterial cells less than the size of a BB. Way less. 100 for some, 10 for others, hundreds of millions for common ones. Infectious dosage does matter.